Hello everyone, myself Dr. Vimal Kumar. I am working as assistant professor in School of Life Sciences, B.S. Abdul Rahman Crescent Institute of Science and Technology. We are going to see about development of vaccine technologies. So this is the overview of this presentation. First, we will be seeing about why India is called a vaccine hub in terms of manufacturing as well as in terms of development. Then we will be trying to understand what are the existing as well as the emerging vaccine technology which are currently available in terms of production as well as in terms of development. And then we will be particularly focusing upon vaccine immunology, where we will be touching upon the basics as well as the findings which are there with respect to the vaccine based responses. And then we will be particularly looking upon nanoparticles based vaccine technologies. So there we will be looking on its benefits as well as its limitations. So based upon that, a few experiments have been designed and performed. So those experimental results will be discussed over there, and finally we will be ending the presentation with a conclusion and future prospects. So why India is called vaccine hub in terms of manufacturing as well as in case of development. So before getting on to that, we can try to understand why vaccines are really important and how it served several numbers of life, and how we have proof of concept that one particular disease it can be specifically taken out from the world. So for that we have the example of a smallpox, so which lead to death around 300 million people in past 2000 years. So in 1980 we terminated this smallpox epidemic due to successful vaccination coverage all around the world. So thereby we, with respect to successful vaccine development as well as the vaccination program both in developed countries and developing countries and underdeveloped countries, we can eliminate a disease from the world. So in case of production, one in every three doses which is administered all around the world, it's been produced in India. In addition to mass manufacturing, India is also involved in creation as well as modifying different kinds of vaccinations indigenously. So these are some of the research institutes as well as your manufacturing companies which are involved in the development of a few of the vaccines which are listed over here, which includes rotavirus, cholera, malaria, as well as pneumococcal based vaccines. So here we have different kinds of prominent institutes like All India Institute of Medical Sciences, the National Institute of Immunology, and ICGB. So we, in addition to that, we also have a different kinds of industries also involved in the development as well as the production process. So this slide it shows the major milestones in the vaccine development all around the world, and then we also can focus upon the Indian based success. So first in 1789 in it started with smallpox, where Edward Jenner he identified the vaccine based things. Then after that we have several kinds of milestones which is there, and then successfully in terms of 2020, so we have a first COVID-19 vaccine by Pfizer BioNTech, and then in recent times we have the booster dose by Pfizer Biotech and all. So before understanding as well as going much into detail, we can see about the vaccine technologies, how it's different from each other's first. So these are the list of vaccine technologies which are currently available. So first we have live attenuated, then inactivated forms and subunit vaccines. First we can see with traditional and subunit vaccines. So Louis Pasteur in 1865 he identified that a weakened form of microorganism can elicit the specific immune responses. For that major principle involved over there is we will be having a set of pathogens, so it can be subjected to repeated culturing, so thereby the virulence factor of the pathogens it will be gone, and thereby instead of causing the disease, based upon the structural components that are present in the pathogens, specific immune responses it can be elicited, or we can chemically modify or inactivate a microorganism, or we can subject it to high heat. Thereby the pathogen, it will be converted from pathogenic, that means disease causing format, to the less harmful form, thereby will be having the immune responses. So this includes some of the examples such as vaccines against the polio as well as the hepatitis, which involves inactivations, whereas in case of attenuations we have influenza, measles, mumps as well as the rubella. So there are success which served the life, which comes with some of the limitations. The first one is going to be a danger of spontaneous reversion to pathogenity. So that means sometimes it's possible that this form, it can be converted into this form. So that means due to the nature of the microorganisms as well as the method which is involved in the preparations, this thing, in addition to eliciting a specific immune response, it can cause the disease by itself. Then the another thing is like some all the diseases we cannot apply this principle over there. For example, in case of cancer, in most of the cases no pathogen is involved over there, so we cannot inactivate or attenuate it. And most importantly, like large kind of vaccines which are first development, we can't give it to children immediately. Why? Because is that they are immune compromised individuals, which might lead to large number of complications. Thereby, after a set of clinical trials and the drawbacks, the iterative process involved that removing the harmful components, and then only it will be given to the child. Then we have special kinds of vaccines, or if it is a non harmful effects means which can be directly given to children. In addition to children, in case of old age people also, we cannot administer all form of the vaccines. So thereby subunit vaccines or keep on evolving. Let's try to understand the principle behind the subunit vaccine. It's nothing but a part of the pathogens, it's going to be taken out. So this part of the antigen, it's going to induce a specific immune response. So in this specificity, it comes with the antigenic component, that means your non-self component from the pathogens. So thereby we can overcome the limitations such as spontaneous pathogenesis, spontaneous pathogenicity, as well as the risk in immune compromised individuals such as babies, old people, or not. So this part of your vaccine, it can be of different kinds of biomolecules. So that means the part of the pathogen, it can be either your DNA or your RNA or protein, or a part of the protein which can be a peptide. So this specific component, either individually it can be given as a part of a vaccine, or together with one or more biomolecules it can be formulated as a vaccine, and then it can be given to an individual. So now we know that in case of the part of the pathogen, that means the biomolecule, it can be used to elicit a disease specific immune responses. But the question is how we can make it as a effective. So that's the thing which is involved. So for that we need to understand the concept of vaccine immunology. So based upon the understandings which are currently available in the research, those concepts has been applied in the development of various technologies. So let's first look on to that what kind of immune cells it's going to be present or elicited or accumulated when an actual pathogen it enters into our body. So let's consider this as your skin. So that can be an epithelial barrier. So the pathogen is outside our skin or the barrier. So it's possible that your pathogen, it can get into this barrier by using its different kinds of invading mechanisms. Thereby first it can recruit our phagocytic cells, which can be a macrophage, which leads to the recruitment of your neutrophil, natural killer cells, as well as dendritic cells. So according to immunological concept, these kind of components, so that means your skin as well as a macrophage, neutrophil, natural killer cells. They are part of your innate immune cells. So your dendritic cell, it serves as a bridge between your innate immunity as well as your adaptive immunity. So we all know the basic difference between your innate immunity as well as your adaptive immunity. So that means it's going to be naturally present, whereas this adaptive immunity, whenever it's going to be elicited, it's going to be keep on adaptive and improving with respect to the type of immune responses that are in need. So this dendritic cells, it can activate your cytotoxic T cells, where your CD8 it's going to be a surface marker, or it can activate your B cells. So it's also possible that your dendritic cells, it can activate your CD4 T helper cells. So to differentiate your innate immunity, it's going to be present from your inborn, as well, whereas in case of adaptive immunity, it's going to keep on developing with respect to the type of the infection which is involved over there. And then in terms of your kinetics, your adaptive immunity, it's going to be slow, whereas this is going to be a fast. This duration of action, it's only going to be present for short, whereas in case of adaptive immunity, it's going to be for a longer time, which comes with the advantage of your memory based thing. So this memory feature of your adaptive immunity can be utilized for the development of the vaccines, where we can specifically activate your B cell as well as your T cells with respect to the receptors which is going to be present over there. So these B cells and T cells have a specific functionality, so which is being mentioned over here. So your B cells, it's going to leads to the differentiation of your plasma cells, whereas in case of your cytotoxic T cells, it's going to act upon your intracellular pathogens. So that means if you are having an infection, means if it is present on the extracellular, means your antibodies, it's going to be bind on the extracellular pathogen and it will be eliminating, whereas your cytotoxic T cells, it's going to act on the reservoir of your infection. So that means your pathogen, it will make utilize of your molecular machineries of your normal cells, and then it's going to be proliferating. So if your cytotoxic T cell, it identifies a specific reservoir, and then if it eliminates from the body, means the infection, it can be eliminated at much effective manner. So in addition to this reservoir, we can also make use of this strategy for eliminating the cancer cells of our body. So this is the functionality of the immune cells. So by applying these concepts, so we have the skin, and then when we are immunizing with the specific vaccines, when the vaccine, if it enters into our body, it leads to the recruitment of different immune cell population. So out of this different immune cell population, the antigen presenting cell, so that means your dendritic cell, that if you specifically modify to activate the B cells, means we are going to have the antibodies. So these antibodies, it's going to specifically act upon your extracellular pathogens that are present in our body. The other possibilities are that if your dendritic cell, if it activates your antigen specific cytotoxic T cells, means it's going to act upon the reservoir of your infections, thereby both your extracellular pathogen as well as your infected cell, that means a reservoir that can be eliminated. Other possibility is that if you are utilizing a tumor specific antigen, means your cancer cell also, it can be eliminated from the body. So the need is crystal clear. We need to have vaccine technologies which elicitate strong cellular responses. So that means your cytotoxic T cells and helper T cells. In addition to that, we also need humoral immune responses. That means your antigen specific antibodies which eliminates our extracellular pathogens that are present during the time of infection. So now we are going to focus only upon your nanoparticles based vaccine technology. So what are these nanoparticles based vaccines is that so we can take a disease specific antigen and then you can formulate in the form of a nano component in the range of 10 nanometer to 100 nanometers. So when these nanoparticles or the nanostructures when it's administered into the body, depending upon the size, the immune cell populations recruited as well as the transported in the in vivo, so that means in the body it's going to be varying. For example, if we have microparticles means those microparticles it will be taken up by your macrophages and then it will be getting into your draining lymph nodes. So we all know that the immune system is going to be consist of lymphatic vessels where we have a lymph, that means the fluid which carries our immune cells. So through those lymphatic vessels your macrophages and the antigen presenting cells it's going to take up those antigen containing particles. Whereas if you are using the nanoparticle means it doesn't need any kind of cells. Directly your nanoparticles it will be draining through the interstitial space that are present in the cells. So directly it's going to getting into your lymph node and then it's going to be taken up by dendritic cells and then it will be activating the specific B cells and T cell responses. So once it is present inside your dendritic cells, then your antigen processing as well as your presentation that matters. So there we can consider two pathways. So based upon the location of your antigenic proteins, the final T cell responses is going to be varying. So in case if your antigen in the form of proteins is present in the cytoplasm means it's going to be processed by the proteosomes and then it will be converted in the form of peptides. So those peptides it's going to be taken up by MHC class 1 components which is present in the endoplasmic reticulum and then it's going to activate your cytotoxic T cells. So when your antigens are present in the form of vesicles, so it's going to be processed by the proteases and then it's going to be loaded onto the MHC complex that are present in the endoplasmic reticulum and then it's going to be transported on the cell surface of your antigen presenting cells which leads to the activation of your helper T cells. So what this slide tells us is that based upon the intracellular delivery location or the intracellular presence on the specific location, the type of T cell responses it's going to be modified. So accordingly we can design a vaccine that where you want your antigen to be present within the cell. So thereby we need to have novel nanoparticles which facilitate the movement of your antigens or the other cargos within the cell. So thereby we are focusing on the cell penetrating peptide which can possibly act as the delivery vehicles. So all from the name itself we can say that the cell penetrating peptides is going to be penetrating into the cells. So these are going to be the shorter forms. So these peptides is going to be crossing our cell membranes. So by getting back to the history, this cell penetrating peptide it's been first identified as a part of HIV virus. So which there in your TAT protein. So this diagram A it represents that based upon the discovery. So we are going to have this pathogen. So this pathogen when it infects your specific normal cells, your TAT protein, so which going to utilize your cell penetrating property. So that means it's going to cross your cell membranes and then it's going to interact with the other normal cells which are present nearby. So thereby this TAT protein it will make a pore formation on the neighboring cells. So this pore formation it facilitates the infection of your HIV. So this is a natural phenomena. So this facilitates the infection in terms of human immunodeficiency virus, whereas this property it's been used for the delivery purpose. So that means in the TAN protein only TAT peptide it will be taken over. So that peptide it can be conjugated either with a drug or with a specific nucleic acid, either by covalent manner or in a non-covalent manner, and then you are going to have a complex between your cell-penetrating peptide along with your protein or nucleic acid based cargo, and then you can deliver your cargo into the cell. So this is what this diagram says, how we can make use of a cell-penetrating feature for different delivery applications. So now, based upon this discovery, several novel designed peptides it's been available with improved functional characteristics depending upon the type of the cargo which has been involved. So based upon the physical as well as the chemical properties, there are several classifications which are being available. So in this case, if it is highly positive means thus cell penetrating peptide, it can be categorized in terms of cationic cell penetrating peptides, whereas if it is going to be highly hydrophobic means then we can categorize as your hydrophobic cell penetrating peptides, when your peptide is containing hydrophobic and hydrophilic component in the peptide, that can be categorized as your amphipathic peptides. So based upon this physical and chemical properties, these peptides it can follow different delivery mechanism as well as the delivery routes within the cells. So when the cargo it's been attached with this specific peptides, rather than direct penetration, a specific endocytic pathways it's been found to be followed. In that case, most of the cargo as well as the peptide it tend to retain within the endosomal vesicles. So if it is retained in the endosomal vesicles means then due to increase in pH, normally there's going to be the degradation of your cargo as well as the peptides within your vesicular components. So when it degrades means the cargo it cannot serve its own purpose. In order to overcome that one, we need to facilitate the transportation of your cargo as well as your cell penetrating peptides to cross the endosomal membrane. So that's why this designer peptide have been utilized to deliver the antigenic cargo into the cytoplasm by overcoming this drawback. So in this case we are going to have an amphipathic cell penetrating peptides. So that means it's going to contain both your hydrophobic as well as your hydrophilic domains. So depending upon the pH, the alpha helicity component of this peptide it's going to be varying. So here as you can see, so you are going to have the arginine which is going to be positively charged as well as your hydrophilic which is going to be there, and other side of your helix we are going to have your hydrophobic components which are leucine as well as the alanine which is going to be present over there. So when this peptides it's present or trapped in the endosome, due to the increase in pH, the helical content of your peptides that also it's going to be increasing, thereby the interaction between your peptide along with your membrane that will be increasing. So when the interaction it increases means which leads to increase of your load on the endosomal membranes. So when the load increases means which naturally leads to the disruption of your membrane, and then instead of getting degraded within the endosomal membranes which been converted into the lysosomes naturally, so instead of going through that route, it will be released into the cytoplasm, so thereby your antigen so that can serve its purpose. So in addition to this alpha helical complex, so this peptide when you are combining it along with your nucleic acid means it can form a nanoparticles. So based upon these designer cell penetrating peptides, in order to have a specific vaccines, we have used the in vitro transcribed mRNA where we use the model antigen ovalbumin. So that means this in vitro transcribed mRNA it encodes for the protein ovalbumin. In the second part, we are going to directly use your ovalbumin antigenic peptides, which been combined along with your sympathetic cell penetrating peptide. So the first comes your particle size analysis. So in this case, you can see that when due to the interaction between a nucleic acid as well as the peptides, it complexes in the range of 100 nanometers. So this NP ratio, it means the presence of the amine group as well as the phosphate groups within each formulation. So with respect to increase in the amount of nucleic acids, then there's going to be your complexation of your nanoparticles which is going to be occurring. So we can see that there's going to be a shift in the formulations due to the differences in the formulation factors. Finally, we can see about your mRNA encapsulation. So that means when you are comparing it with different formulations at NP10, there can see that nearly 100% of mRNA it can be incorporated into the nanoparticles. So therefore, for the further studies, we have used this formulation NP10. So when this formulations, we know that in case of only the peptide, we have this pH dependent membrane disruption properties. In order to make sure even when the complexation occurs, is there any deviation from the pH dependent membrane activity or not? So thereby did the hemolysis assay, as we can nicely see that both in terms of pH 7 as well as in terms of pH 5, that's going to be differences in the hemolytic properties. So then we need to look upon whether the mRNA which we used as a cargo, whether it's functional or not. So that means whether the transcript mRNA it's been translated into protein or not. So thereby we used this green fluorescent protein encoding mRNA. So through that the nanoparticle it can be used, and then when it is delivered, then it will be able to undergo translation and it needs to express the specific protein. So thereby we can say that the nanoparticle which is being formulated are able to retain the properties as well as whatever the cargo which it carries, it's not getting affected due to the complexation. So then we need to do the nucleoside modification. So natural uridine it's been modified into pseudo-uridine, and in case of citidine we can use this methyl citidine. So when you're modifying these things means unwanted immune elicitation or recognition by the other innate receptors that can be minimized, thereby your mRNA it can encode for specific protein at the higher rate. So that's what this slide says. A nucleoside modification, it enhances your mRNA expressions by lowering your innate immune expressions. So we highly it's been used in the concept of gene therapy, such as your erythropoietin as well as your stem cells based studies. So whereas in case of your T cell immunity, we have nicely found that this modified mRNA has had the beneficial outcome when comparing to your unmodified mRNA. So the statistics it shows that the values are highly significant when you are using the unmodified as well as a modified in the form of nanoparticles. Whereas if you are using only the in vitro transcribed mRNA, is the elicitation of T cells are much more lesser, whereas only if we are complexing with the peptide and then making as a particle format, the T cell responses it's going to be pretty much higher. So to summarize, so we had this antigen coding mRNA. So with respect to the specific disease which you're going to work on, you can choose this specific mRNA which can be transcribed in the in vitro level without any need of cells. So this in vitro transcribed mRNA it's been combined with the designer peptides. So in this case we have used the cationic amphipathic peptides. So both combines together and forms a nanoparticles lesser than 100 nanometers nanometers. So when it is cultured along with your antigen presenting cells, we nicely saw that the pH dependent cell disruption sits there. So there, due to the disruption of your cell membrane in the endosomal compartment, this mRNA it's going to be released out of that, thereby the antigen it's been converted from mRNA format to the protein format, and then this antigen it's going to be processed and presented upon its cell surface, which leads to the elicitation of antigen specific T cell responses. So next we are going to look upon your peptide based antigens. So where we are going to combine your specific disease specific antigen along with your carrier peptide. So there we are using a nucleic acid which doesn't code for any kind of antigen. So when you are complexing these two things together, this transmission and electron microscopy image nicely shows that it forms a spherical particles without any aggregation. So when it's been incubated along with your dendritic cells, these things it's going to be getting into the cells. So that means your cargo it's been getting delivered within the cells. So that's what this slide shows to us. So when it's been antigen it's been combined along with your specific carrier peptide, so we compared it along with only your peptide and then we complexed along with your carrier peptide. So in this case we have the first formulation, so where you have only RALA MHC1 epitope. So that means your carrier peptide is combined along with your antigen, whereas in the second one you only have antigen, and then the third one only have your nucleic acid. So in case of your mice, first one which is not included with any of this doesn't have any immune responses, whereas when you included your carrier peptide along with your antigenic peptide and nucleic acid, that's going to be a higher immune responses when you are comparing it with the other cases. That means your only antigen as well as your antigen along with your carrier peptide. So that means these complexation of a nanoparticle it elicits a high T cell responses. So this is in case of a cytotoxic T cells. Next we looked upon your helper T cells. So the same study it's been performed, and then when we looked upon your antibody responses, these nanoparticles they are able to produce your antibody responses. So upon further immunization with your recombinant protein, that responses it can be further increased to different level. So thereby we can have a different kinds of immune responses with combination with only your peptide and peptide along with a nucleic acid, or we can further boost the immune responses using a specific recombinant protein. So from the model antigen, then we shifted on to the melanocyte lineage specific antigen GP100. So when this specific peptides it's been coupled along with the specific nucleic acid, those also are able to form as a nanoparticle and induce the T cell responses, and those T cell responses are seen to be functional. So to conclude, like we are having a vaccine technology so which can elicitate both your cellular as well as your humoral immune responses, so whereby your cytotoxic T cell, helper T cells as well as your antibody responses, everything it can be co-induced, but we need to specific about this antigen that's used depending upon the specific application. So if it is focused only on their cancer, then we need to use a specific neoantigen or the other antigen which are involved in a personalized manner. Whereas if you are looking onto the infection, means if we use only the antibody elicitation, means only the pathogens that are present out of the cells that are focused. Whereas if we are having a cytotoxic T cell responses that are specific to the antigen, means then the reservoir of the infection by itself that can be eliminated. So the most advantage of this one is we don't need any kind of cells, thereby the regulatory approval process as well as the production in terms of different batches, that's going to be pretty much faster and it's going to be more cost effective. So thereby if you have a pandemic or epidemic means immediately we can counteract with this technologies. The future other study is that we can also use a specific sustainable biomaterials which are naturally existing, and it can be fine-tuned with the chemical modification, thereby additional immune responses in terms of innate or with respect to the specific disease, the adaptive immune responses, those can be synergetically induced in the patients. So thank you. These are the collaborators.